blood cultures are taken by clinicians when the patient's signs and symptoms suggest the possibility of bacteremia or septicemia. Bacteremia is the presence of bacteria in the bloodstream. Septicemia is the presence and multiplication of bacteria in the bloodstream, together with the symptoms of systemic infection, which may include high fever, chills and rigors, with the potential for systemic shock, multi-organ failure, and death. Bacteria and other pathogens can get into the bloodstream in various ways, most commonly from an infected site, such as an abscess or tissue, or from an infected organ, for example, the kidneys, or from a colonized intravascular line that is inserted into the patient. A blood culture involves aseptically taking a sample of blood, usually 5 to 10 ml, from a patient's vein, and inoculating it into commercial blood culture bottles. These particular bottles are Becton Dickinson Bactec FX bottles. Several media types are available, but the most common ones used are aerobic and anaerobic media bottles. The media contains nutrients and growth factors to support the growth of the organisms. They also contain resin beads to help to neutralize the effects of antibiotics and neutrophils and other potential inhibitors that may be in the blood. On taking the blood culture, the sample should be immediately transported to the laboratory, and on arrival, it is barcoded onto the blood culture analyzer. The analyzer is an integrated piece of equipment consisting of an incubator at 35 degrees to allow optimum bacterial growth, an agitator, the bottle racks rock continuously, and a growth detector system inbuilt into it. Blood culture bottles are loaded onto the analyzer and incubated for up to 5 days. When microorganisms are present in the blood culture, they metabolize the nutrients within the culture medium and release carbon dioxide into the medium. A dye sensor in the bottom of the vial reacts with the carbon dioxide. This reaction modulates the amount of light absorbed by a fluorescent material in the sensor. Each blood culture station in the analyzer contains a light-emitting diode photodetector, which measures the level of fluorescence every 10 minutes. A negative vial produces a linear graph as demonstrated. A positive vial produces an exponential curve graph as can be seen. Once the fluorescence reaches a specific threshold, the positive indicator light flags on the machine and the biomedical scientist needs to remove the bottle to find out what is potentially causing the bloodstream infection. We cannot predict what might be present in a patient's blood culture, and therefore, in this setting, all manipulations of positive blood cultures need to take place in special containment equipment to protect the lab staff, and in this case, a Class 1 safety cabinet is used. A sample of blood is removed from the positive bottle inside the Class 1 safety cabinet using a venting device. A microscope slide smear of the blood is made and heat fixed prior to gram staining. The gram stain slide is then examined under a light microscope at a ×1000 magnification. The most common organisms found in blood cultures include Staphylococcus aureus, Escherichia coli and other coliform bacteria, Streptococcus pneumoniae, Enterococcus species, Pseudomonas aeruginosa, and Candida albicans. This particular positive blood culture shows gram-positive cocci in pairs, diplococci. On the basis of this gram stain, the biomedical scientist can also perform a range of direct identification tests on the blood to aid with the presumptive day 1 diagnosis. In this case, the gram stain showed gram-positive diplococci, and the patient clinical details were pneumonia. Therefore, this organism could be Streptococcus pneumoniae. To help with the presumptive identification of this organism, a direct latex test that detects the presence of pneumococcal antigen in the blood using specific antibody-coated latex particles can be carried out. Control and test latex particles are mixed with a drop of the blood. In this case, the test latex agglutinates with the blood and the control latex remains clear, thus indicating the presence of the pneumococcus, Streptococcus pneumoniae. Culture of the blood is made onto agar culture media, including chocolate agar incubated at 37 in carbon dioxide and a blood agar incubating anaerobically at 37. To help with the identity of this organism, a paper disc containing a chemical called opticin is placed onto the blood agar plate. Direct antibiotic disc susceptibility tests are also performed on the positive blood culture. Either a direct inoculum of the blood is used or a diluted inoculum, dependent on the organism. This is spread onto a sensitivity testing agar using a rotary plater to create an even semi-confluent inoculum lawn. Filter paper discs that contain specific antibiotics are applied to the sensitivity plate using a disc dispenser, and during incubation the organism will grow, and at the same time the antibiotics will diffuse out of the discs, and sensitive bacteria will be inhibited by the antibiotic and not grow around the discs, while resistant bacteria will not be affected by the antibiotic and grow up to the disc. Thus, the culture and sensitivity plates are incubated for between 18 and 24 hours and then examined. The aim for the scientists working on the blood culture bench is to have a culture result, a presumptive identification of the pathogen, and an antibiotic susceptibility result within 24 hours of the bottle becoming positive on the machine. As indicated, this patient's blood culture grew a Streptococcus pneumoniae. This plate shows alpha hemolytic colonies on blood agar that are sensitive to opticin, and the sensitivity plate shows the isolate to be fully sensitive to the antibiotics tested.